So I'm going to take the biology for engineers and biodiversity. I'm going to take plant system, basic concept of plant growth and nutrition. Introduction. Let us see the introduction of plant system. All cells of a plant develop from the zygote. Zygote produce a number of cells which organize into tissues and organs. Development is the sum of two processes, growth and differentiation. During the process of development, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits, seeds, and eventually they will die. Growth. A growth may be defined as the irreversible payment, increase in size, volume, or mass of a cell or organ or whole organism. Growth is one of the fundamental characteristics of the living being. It is accompanied by metabolic process, that is anabolic or catabolic process that occurs at the expense of energy. For example, expansion of a leaf, elongation of stem, etc. There are four main characteristics of growth: cellular growth and cellular division, cellular expansion and cellular differentiation. Growth rate can be defined as the increased growth per unit time. The rate of growth can be expressed mathematically, that is arithmetic growth and geometric growth. So arithmetic growth, after mitotic cell, only daughter cell continues to divide while the other differentiator mature. That is root elongating at the constant rate. Mathematically it is expressed as LT is equal to L0 plus RT. Geometric growth. Initial growth is very slow, that is followed by the rapid increase in growth and followed by a phase where growth slows down. For example, all cells, tissues or organs shows this type of growth. If one plots the parameter of the growth against time, it will be the typical S-curve. The exponential growth can be expressed as W1 is equal to W0 ERT. Next one, differentiation. So differentiation is the process in which a less specialized cell become a more specialized cell. That is what the differentiation. The cell derived from the militants and cambium differentiate and mature to perform the specific function, which is termed as a differentiation. So cell undergoes structural changes during differentiation. Changes take place both in the cell walls and protoplasm. Cell lose their protoplasm during the formation of tracheiry elements. Plants develop a strong, elastic and ligocellular cellular secondary cell walls to carry water to long distance even under extreme tension. De-differentiation. De-differentiation is an important biological phenomenon where cell regress from the specialized function to a simpler state, reminiscent of the stem cells. The undividable differentiated cell sometimes regains the power of division. This process is known as de-differentiation. De-differentiation is a common process in plants during secondary growth and in wound healing mechanism. Redifferentiation. The de-differentiated cell can be divided and produce new cells. New cells produced again lose the power of division and become the part of the permanent tissue, which is called redifferentiation, that is formation of tumor cells. Development. Development is a term that includes all changes that an organism goes through during this life cycle, from germination of a seed to senescence, that is meristemic cell, plasmadermic cell, differentiation and mature cell. At last it will go to expansion or maturation. Next, plasticity on heterophyll. Heterophyll. The ability of the plant to follow different pathways and produce different structure in response to environment and phases of life. In cotton and coriander plants, the leaf of juvenile plant in different shape from those in mature plants. Heterophyll, the phenomenon of appearance of different forms of leaf on the same plant, is called heterophyll. Plant growth regulators. Plant growth regulators are chemical substances that influence the growth and differentiation of the plant cells, tissues and organs. Plant growth regulators function as the chemical messengers for intercellular communication. They work together and coordinating the growth and development of cells. They could be indole compounds, that is adiene derivatives of carotenoids and therapeutics or gases. So phytohormones. There are five types of phytohormones: auxins, gibberellins, psychotins, abscisic acid, and ethylene. And PGR, that is phytocarbons, is again classified into growth promoters, growth inhibitors, auxins, gibberellins, psychotins, abscisic acid, and ethylene. Let us see what is auxins. Auxins were discovered by Charles Darwin and Francis Darwin. FW, that is Charles Darwin and Francis Darwin went as isolated from the tips of caulopaths of oat seedlings. When developed Avena curvature test of biodiversity of auxins. Auxins are the group of naturally occurring and artificially synthesized hormones. They play an important role in the regulation of plant growth. Auxins were initially isolated from the human urine. Auxin means enlarge or increase. Next, psychological effects of auxins: promote apical dominance, initiating rooting system in stem cuttings, promote flowerings, prevent fruit or leaf at early stage, promote the aggregation of mature leaves, and induce parthenocarpy, used as a herbicide, controls xylem and differentiation. Gibberellins. Gibberellin is a plant hormone regulating key process in a plant. Many of them are of significant agricultural importance, such as seed germination, root and shoot elongation, flowering, and fruit patterning. Gibberellins are the plant growth regulators that facilitate cell elongation, help the plants to grow taller. They also play a major role in germination and elongation of stem, fruit ripening, and flowering. Gibberellins. Also, gibberellins was invented by the Japanese scientist, named as Gibberella fujikuri. Gibberellins are isolated from the infected rice seedlings when treated as a sterling filtrants of fungus. Elongation of intact stems, draft shoots, bottling in roastily plants overcome the natural dominancy of buds, tubers, and seeds. Seed germination, induce parthenocarpy, promote flowering, vernalization. Psychotins. Skunk and Miller discovered the psychotins. Skunk and Miller crystallized the psychotins, promoting active substance named in chitin or from coconut milk. Induce cell division, delay the senescence of leaves and other organs, promote lateral birth, increase cell expansion in dicot and in leaves, promotes chlorophyll development and chlorophyll synthesis. Ethylene. Cousins conform with the release of volatile substance from ripened oranges that hasten the ripening of stored and unripened bananas. The volatile substance was identified as ethylene, promotes senescence and application, inhibits elongation of stems and roots, induce fruits ripening, break seed and bud dorfany, initiate germination in peanut seeds, spotting of potato tubers, promotes root growth and root hair formation, and initiate flowering for synchronizing fruit set pineapples. So thank you all. Let us see the next class.